All right, thanks for joining me today. Antibiotic resistance is such a great topic in the classroom because you can cover natural selection while also tackling a real world health problem. The lab I'm going to be demoing today, the BioBits Antibiotic Resistance Lab, will allow your students to do a hands-on lab exploring antibiotic resistance, but without having to grow bacteria or deal with antibiotic-resistant bacteria cells. But first, a bit about us. My name is Dr. Ali Huang, and I'm one of the team members at Mini PCR Bio. For those of you new to us, we're a biotech company that makes easy to use and portable versions of traditional molecular biology equipment, such as PCR or gel electrophoresis machines. Both teachers like you and researchers use our equipment in the classroom or in the field, like in the Amazon or out foraging for mushrooms, as shown here, and even in public health settings to monitor viral outbreaks. And one of the coolest examples of our experiment being used in the field is actually in space to run experiments on the International Space Station. If you're interested in that, check out our Genes in Space competition for more information. All right, when we go back to classroom use, however, we have a huge catalog of learning labs that help teach a variety of biology concepts. To us, it's really important to emphasize that biotechnology is a tool and not just a topic on its own. And our curriculum uses biotechnology as a tool to explore fundamental concepts and core ideas. This allows students to have a better context for how biotech can actually be used and involves them in investigating phenomena rather than just learning techniques. For example, our Missy Baker Missing Lab uses PCR and gel electrophoresis to explore how genetic differences can cause diseases at the cellular level, like cystic fibrosis. So that's just one example. But before we get into the details of the BioBits Antibiotic Resistance Lab, let's take a step back and first go over some background info on antibiotics. Many of us have probably taken antibiotics before to treat or prevent common bacterial infections, and we probably take antibiotics for granted, but once those bacterial infections were deadly. But since the first antibiotic, penicillin, was discovered in 1928 by Alexander Fleming, pictured here, those bacterial infections became very easy to treat, and it's actually estimated that millions of lives are saved each year by antibiotics. So it's not an exaggeration when I say that antibiotics are one of the greatest medical advances of the 20th century. However, nearly 100 years after that initial discovery, today more and more bacteria are becoming resistant to antibiotics. In fact, it's estimated that 1 million people die a year worldwide from antibiotic-resistant bacterial infections that antibiotics can no longer treat. And indicated by this 2016 study, this number could increase to 10 million deaths per year by the year 2050, which would overtake other causes of death such as cancer. So as you can see, this is obviously something that health officials and a lot of other people are very concerned about in terms of global health. So what can be done? One answer is to use antibiotics more appropriately. The rampant spread of antibiotic resistance is largely due to our misuse of antibiotics, whether it's hospitals overprescribing them or patients not taking them correctly. Today's lab, however, is not going to be focused on that, but I'll mention resources in the end in case this is a topic you would like to cover. Instead, our focus today is going to be how antibiotics and antibiotic resistance works at the mechanistic level, because that is important for antibiotic discovery. New antibiotics can be discovered to fight the bacteria that are resistant to our current antibiotics. Antibiotics, in fact, are mostly discovered in nature, not created, like how Alexander Fleming did with penicillin from mold. These days, scientists have the ability to screen really large collections of potential antibiotics to try to find new ones. And in order to do that effectively, we need to understand how both antibiotics and antibiotic resistance works, which scientists refer to as the mechanism of action. So we're going to get down to the nitty-gritty molecular details now, and when we get down to these biochemical details, an antibiotic is a small chemical molecule that can kill bacteria. And there are many different kinds of antibiotics, and each antibiotic has both a specific target in the bacteria as well as a unique method it uses to disrupt that cellular target. For example, in today's lab, we're going to be using 2 antibiotics, both of which specifically target the bacteria ribosomes involved in protein synthesis. So like a ribosome, like I'm picturing here. Once we get into the lab, I'll describe the more unique methods each of these different antibiotics have to target the bacteria ribosomes. And I keep specifying bacterial ribosomes because that's another important element to antibiotics. They all only target these mechanisms in bacteria, not human cells. This makes antibiotics safe for us to take. If all of this is too advanced for your students, you can distill all this information down to a simple takeaway, which is just that each antibiotic targets bacteria in a very specific way. All right, so moving on to antibiotic resistance. How do bacteria become resistant to antibiotics? So let's first define what we mean by resistance. Bacteria are resistant to antibiotics when they can survive in the presence of it, like I'm demonstrating here. The surviving bacteria is more likely to survive and reproduce, and over time, we will end up with more antibiotic-resistant bacteria. While this is a really great example of natural selection, antibiotic resistance is not so great in terms of human health. As more bacteria become resistant to more and more antibiotics, it's going to be harder and harder to treat these infections. Again, let's zoom down to the molecular level to see how all this happens. So bacteria that are resistant are able to produce proteins that protect them against the mechanisms of antibiotics. Just like how antibiotics can kill bacteria in lots of different ways, proteins that can protect bacteria from antibiotics can also work in lots of different ways. There are proteins that pump antibiotics out of the bacteria. There are proteins that chemically modify the antibiotics to make them ineffective. Like antibiotics, these antibiotic-resistant proteins also have specific targets and unique ways of targeting them. Now, the instructions to make these proteins, like all proteins, any proteins, are encoded in the bacteria's DNA. So therefore, it's the genetic info that gives bacteria resistance to antibiotics. And we call the part of the DNA that encodes the protein responsible for resistance the antibiotic resistance gene. All right, so to sum that all up, this is a regular bacteria with no resistance to any antibiotics. When it's exposed to antibiotics, it dies because the antibiotic disrupts some essential cellular process. Each antibiotic does it differently, but again, as a reminder for today, we'll be looking at antibiotics that disrupt ribosome function in protein synthesis. Now, these bacteria right here have an antibiotic resistance gene, pictured in green, which encodes for a protein that provides protection against a specific antibiotic. In this case, because these bacteria have antibiotic resistance that protects them from the specific yellow antibiotic, they'll be able to survive. But because each antibiotic resistance gene provides resistance to a specific antibiotic, if I expose this bacteria to a different antibiotic, it dies because its antibiotic resistant gene only gave it protection against the yellow antibiotic, not the red one. Again, I know that was a lot about antibiotic resistance, so if you only remember one thing, it's that antibiotic resistance genes confer protection against specific antibiotics. All right, so your students will be able to explore these key takeaways that I just talked about antibiotics and antibiotic resistance with our new BioBits Antibiotic Resistance Lab. This is an authentic, hands-on lab that demonstrates these different mechanisms without having to culture any cells. Briefly, your students will test 2 different antibiotics and 1 resistance gene using our BioBits cell-free system that requires very little equipment. And after doing the lab, they hopefully will understand the mechanisms of antibiotics and antibiotic resistance and how they work and how specific they are. All right, so your goal in this lab is going to be twofold. You want to confirm that both antibiotics you're using do block translation and protein synthesis. And this will demonstrate the first main takeaway that I talked about, that different antibiotics target bacteria in specific ways. And 2, you want to determine which of the 2 antibiotics is susceptible to the resistance gene. This will demonstrate the second main takeaway, that antibiotic resistance genes confer protection against specific antibiotics. All right. So, antibiotics and antibiotic resistance are usually studied in live bacteria cells. For example, you might expose a bacteria culture to an antibiotic to see which cells survive, and then study those cells to see what genes allowed them to survive. But that can be difficult to do in a classroom. Not only does it take a lot of time and require specialized equipment, there also could be biosafety lab concerns, especially when you're playing around with potentially antibiotic-resistant bacteria. So as an alternative, today, we're going to be using BioBits, which is a cell-free system that contains all the molecular machinery you need to make proteins. For example, like you see in this picture here, like the ribosomes that we'll be focusing on today. So they contain these ribosomes. Now, if we wanted to look at antibiotics that targeted cell wall synthesis or DNA replication, we would have to use bacteria cells because the cell-free system doesn't contain those elements. But because we are looking at antibiotics specific to protein synthesis, we can use this simpler and safer model that allows us to observe those mechanisms. So getting into a little more detail about how BioBits works. BioBits will come freeze-dried at these little pellets. I'm going to show them on camera in a little bit. And again, these pellets contain all of the protein synthesis machinery and materials. The students are going to add DNA to these BioBits to rehydrate the pellet. And again, since this DNA contains instructions to make the proteins, and the BioBits contains the machinery to make the protein, the system will make whatever the protein encodes for, or make whatever protein that the gene encodes for. For today's lab, we'll use the system to generate both a protein that demonstrates protein synthesis is working, as well as an antibiotic resistance protein that can protect the system from ribosome targeting antibiotics. Again, if that's all too complicated, you can just tell your students that the BioBits system is a model that can be used to specifically test if the antibiotics are blocking protein synthesis. So in today's lab, we're going to be making fluorescent proteins, like these pictured here, that demonstrates if protein synthesis is working. Fluorescent proteins are one of the only proteins that you can easily see and identify without lots of expensive equipment, and it's really important for this lab to be able to tell if protein synthesis is occurring in the presence of antibiotics. For context, fluorescent proteins are naturally found in organisms like jellyfish, and scientists often use fluorescent proteins to visualize cellular processes and structures that they normally can't see to help them understand what's going on. And that's exactly how we'll be using these fluorescent proteins today. So to visualize these fluorescent proteins, which fluoresce when exposed to blue light, we're going to be using our P51. Our P51 molecular viewer is an inexpensive cardboard handheld blue light transilluminator, which you can use to visualize if there's anything fluorescent in your tubes. And we call it the P51. It's named after Rosalind Franklin's Photo 51, because Photo 51 was a way to understand what was happening on the molecular level in a visual way, just like what we're going to be doing today. And when I get started doing the lab, you'll actually be able to see my P51 in action. So the lab itself is very straightforward and quick. In the first class period, you'll have your students set up their reactions, which takes about 30 to 45 minutes, so it fits in nicely in a class period. And in this lab, we actually have you split the lab groups where half of them are going to test one of the antibiotics and the other is going to test the other. In the end, everybody in the class is going to pull their results all together to make conclusions together. Then when they come back to the next class period, after they're all done setting up their reactions, they can observe their final results, and that's when they pull all their results together and compare. Because we're using a cell-free system, this lab does not require a lot of equipment. In fact, as you can see, I'm just in my office, and all I'm going to use is a micropipette, a 2 to 20 microliter micropipette and tips, as well as that P51 blue light transilluminator. All right, so at this point, I am going to change my screen up so I'm a little bigger. So you're going to be adding the reagents in 2 steps. And the first step is to add either water or kinase DNA to the BioBits pellets. And during the incubation, I will explain what I mean by all of this, so just hold tight. In the meantime, I'm going to show you how easy it is to use BioBits. All right, so I have my micropipette here. I'm going to set it to 3 microliters because that's the only volume that I'm going to be using. And yes, it's a good idea to have your students wear gloves as they're doing the lab, but there's nothing really toxic about this lab, so I'm just going to go gloveless today, but tell your students to wear gloves. All right, so these 4 tubes here, I've already labeled tubes 1, 2, 3, 4. I don't know if it's showing up on camera very well, but I promise you that there are 4 little pellets sitting at the bottom of my tubes. Those are the BioBits pellets that, again, contain not only the ribosomes that we're going to be targeting, but also the ribosomes and the rest of the molecular machinery that we're going to use today to make the red fluorescent protein. All right, so all I'm going to do is I'm going to open these tubes, and we're going to add the reagents as what's shown on this chart. So let's see, I have my water tube right here. I'm going to add water to tubes 1, 2, and 3, and then kinase DNA to tube 4. So all I'm doing is just pipetting up 3 microliters just like that. And I'm just adding it to my tubes like that. Easy peasy. All right, so I'm going to do the rest of the reaction on my table here so I can go a little faster. And again, as I am adding all these reagents, after we add all these reagents, I will go over exactly what the lab setup looks like. Okay, there we go. I've added reagents to all 4 of my tubes. I'm just going to tap them on the table so all the liquid goes to the bottom. Sometimes if they get stuck on the side of the tubes, you just have to use your pipette to wash them off. There we go. All right, so I'm going to pop the caps back on. And as you can see in the camera, hopefully you can tell they have been rehydrated. So yeah, there we go. You can see it a little better there. So they all have 3 microliters inside there. And all I'm going to do is just leave this on my desk right here for 10 minutes. And then during these 10 minutes, again, you just have to incubate these at room temperature. I'm going to go ahead and explain what the actual setup of this lab is. All right, so I'm going to switch back to slide view. There we go. All right, so to some of our BioBits pellets, I'll be adding RFP DNA, which is DNA that encodes instructions for making a red fluorescent protein, which I'm going to refer to as RFP. RFP equals red fluorescent protein. If, say, we transform a live bacteria culture with this RFP DNA, the bacteria would make RFP and glow red. Similarly, in our model we're using here, with the added RFP DNA and the protein machinery in BioBits, is shown here, RFP will be made and we'll be able to see that glow red in our tube. So if we see glowing red, that means that translation was successful, the ribosomes are still working, and protein synthesis is functional in our BioBits model. Again, the important takeaway for your students to remember is red means that successful protein synthesis happened, and this models that bacteria are alive. Again, we're not using real bacteria in this lab, but the cell-free system models what the bacteria would behave like. All right, now to some of our tubes, we'll also be adding antibiotics. The student lab groups will add either kanamycin or streptomycin, depending on what you assign them. Both of these antibiotics block translation in bacteria, specifically by binding to the ribosomes. If you're curious, even more specifically, kanamycin disrupts the binding of mRNA to the ribosomes, while streptomycin disrupts the binding of tRNAs to the ribosomes. Again, a great example of how different antibiotics function in very different and specific ways, even if their target in the end is the same. If we add either of these antibiotics to a live bacteria culture, the bacteria will not be able to make proteins and will die. And since BioBits contains bacteria ribosomes, we can also use them to test the functions of these antibiotics. So when we add the antibiotic, we expect to see translation inhibited, resulting in no RFP. This means we're not going to see any red fluorescence, and that will tell us that there was unsuccessful translation due to the ribosomes being inhibited and that the antibiotic added was successful in blocking protein synthesis. Again, the takeaway, if this is hard to follow, is that no red means no protein synthesis, and this models bacteria that are dead from antibiotics being added. Finally, the antibiotic resistance gene we're testing is called kinase DNA, and kinase DNA encodes a protein known as aminoglycosidase kinase. I'm just going to call it kinase for short, even though there's lots of different kinases, but the purpose of this lab, we're just going to refer to this enzyme as kinase. This kinase can provide resistance to some antibiotics, and we're going to find out if this includes kanamycin and or streptomycin. Now, going back to the live bacteria culture analogy, if the bacteria in that culture had this resistance gene in their DNA, it would be able to produce the kinase and have potential protection against certain antibiotics. Again, since red equals translation worked and no red equals translation didn't work, we'll use the readout of the reaction to determine whether or not this antibiotic resistance gene was able to confer resistance to the added antibiotic. So, bottom line, if you added antibiotics but your tube still glows red, then that means for the added resistance gene, that protected protein synthesis from the antibiotic, just like if bacteria were able to survive even if an antibiotic was added. All right, so let's make some predictions together, and I'll go over tube by tube what is being added to each reaction. So in tube 1, we added just water to our BioBits pellets. Do we expect to see RFP if we added no DNA to the model? Yes or no? I'll give you guys a few seconds to respond in the chat. Our moderators are in there, so if you have any questions, by the way, feel free to pop them in the chat, and one of us will answer your question. All right, so that's tube 1. In tube 2, we added RFP DNA to the BioBits model. Do we expect to see RFP, the protein, if we added DNA that encodes for RFP here? It helps to think about what would happen in a live bacteria culture like I referenced before, and then apply it to the BioBits cell-free model here. So again, put what you think in the chat. In tube 3, we added the RFP DNA as well as one of the antibiotics to our BioBits pellets. Again, like I mentioned before, both of these added antibiotics, either kanamycin or streptomycin, depending on which one you're doing, will inhibit the ribosomes. So based on that info, will we see RFP? Yes or no, and why? And finally, in tube 4, we're going to add everything. We're going to add the RFP DNA, we're going to add one of the antibiotics, and we're going to add the antibiotic resistance gene. And since we don't know which antibiotic the antibiotic resistance gene confers resistance to, we don't know if we're going to see RFP, right? So this is something we can't really answer until the end. If we see RFP when the antibiotic resistance gene and the antibiotic are added together, what does that mean? Write it in the chat. And on the flip side, if we don't see RFP when the antibiotic resistance gene and the antibiotic are added together, what does that mean? Again, you can explain in the chat or just think about it. All right? So that is how the lab is set up, reaction by reaction. All right, so if we get back to the tubes, again, this is a reminder of what I have already added. And just as a little pop quiz, does anyone know why we added the kinase DNA first, waited 10 minutes before I'm going to go on and add everything else? If you think you know, put it in the chat. Well, here's a reminder that these antibiotics inhibit translation, all translation, including that of the kinase, right? So if we added everything at once, the system wouldn't be able to make the kinase in time to protect itself against the antibiotic with. So we give the kinase a little bit of a head start by adding it first. All right, so I'm actually going to add the rest of the reagents now. I don't know if it's been quite 10 minutes, but for the purposes of demoing, that will be okay. So these are my tubes that I just added to. And I'm just going to do a set of 4, assuming that I'm the lab group that got kanamycin assigned to it, okay? So I'm just only going to set up for kanamycin, but I also will show you results with streptomycin and talk through that as well. All right, so for always adding these reagents, again, it's just 3 microliters with your micropipette. Tube number 1 is just going to get water again. So I'm going to add that there. Now tube number 2 is going to get the RFP DNA. So let me switch my tubes here. I'm going to add that. Tube 3 is going to get RFP DNA and one of the antibiotics. And just to make it easier for your students to pipette so there's no mistake, we've actually included a pre-mix of the antibiotic and the RFP DNA mixed together. So you only have to add just one pipetting action to that tube. So tube 3 gets that. And then tube 4 gets the same, but remember in tube 4, in the first part, that got kinase DNA instead of water. So that's the difference there. All right, tubes are going everywhere on my desk. That's okay. And I'm just gonna shake things down. So now I have 6 microliters total in all of my tubes. You can see here. Yep, right there. All right, so once you've added everything, you can actually take an initial peek in your P51. And so this is the P51. As you can see, to use the P51, you just open it up. And I'm going to pop my tubes in the holes in the top there. And I'm going to turn it on. And it's powered by a 9-volt battery. We also have ones that you can plug in. And when you turn on the blue light, you can see the blue light shines up at the tubes. And if there's anything fluorescent in the tubes, I can actually see it through this little acrylic window. However, right now you shouldn't see anything yet. Again, this is all just baseline. You'll get your final reaction after an overnight incubation at room temperature. Since you probably don't want to wait here with me until tomorrow, I'm just going to show you the tubes that I set up yesterday, cooking show style. All right, so here are the tubes that I set up yesterday. Let me put them in. Let me get the camera to catch them. All right, so on the left, I hope it's my left. So let's use my finger. Here, these ones, I think my camera mirrors it, so I'm just making sure. So here we have tubes 1, 2, 3, 4 for the set of kanamycin, and the next 4, 1, 2, 3, 4, are tubes 1 through 4 for streptomycin. Interesting, right? All right, so I'm going to go back to my slides now and review the results. So we can actually go through tube by tube and see what's going on. But again, this is something like what you students will see, bright red fluorescent protein. Pretty cool, right? All right, turn that off. And I'm going to switch back to my slides a little bigger. All right, so let's recap what we just saw. Again, tube 1 just had water, so we saw nothing for both sets as pictured here. Tube 2 had RFP DNA, and we do see RFP for both sets. We do see that glowing red. Tube 3 had RFP DNA and either kanamycin on the left side of the screen or streptomycin on the right side of the screen. And looks like we didn't see anything in those tubes. And then tube 4 had the antibiotic, had the RFP DNA, but also had the kinase DNA, the antibiotic resistance gene added. And we see RFP in the kanamycin set, but nothing in the streptomycin set. So why don't you guys start chatting in the chat to talk about what do you think this means. But I am going to go through it tube by tube at the molecular level so we're all on the same page, starting with tube 1. Tube 1, we saw nothing. And because this tube only had water, there's obviously no translation because no DNA was ever added. And the reason we have this tube is to use as a negative control and as a baseline to compare our other tubes to. All right, so in tube 2, we saw red. If we see red, this means we have RFP, we have our red fluorescent protein. This tells us that, as expected, translation did work. We got RFP from the RFP DNA added. And again, the purpose of this tube is our positive control. What does successful RFP and translation look like? Again, this tube, if we go back to a real live bacteria culture, this represents that bacteria are alive and functioning. In tube 3, we do not see anything. This tells us that no RFP was made. Since we added antibiotics to this tube, we can conclude that translation failed, and it was due to the antibiotics inhibiting the ribosomes, causing no protein synthesis of the RFP. And this is true for both lab groups, whether or not they used kanamycin or they used streptomycin. As a reminder, kanamycin blocks mRNA from the ribosomes, while streptomycin blocks tRNA from the ribosomes. But despite the different mechanisms, both have the same outcome, no protein synthesis. Since this shows the function of both kanamycin and streptomycin, this is actually kind of a form of a positive control for the antibiotics. In terms of the real world, if you were in a lab working with actual bacteria, this tube here will represent bacteria that died from either kanamycin or streptomycin, inhibiting their ribosome and protein synthesis. All right, so let's get to tube 4. Let's start with the lab group that used streptomycin. So in this tube, we saw no RFP. So this tells us that translation didn't work and protein synthesis did not happen. That means even though we added this kinase DNA, which made the kinase protein, this did nothing to prevent the streptomycin from inhibiting the ribosome. Therefore, we can conclude that the antibiotic resistance gene, this kinase DNA that we added, does not confer resistance to strep. Again, lack of translation, lack of fluorescence tells us that the antibiotic is working, so therefore the antibiotic resistance gene we added doesn't protect the system against that antibiotic. Again, going back to our bacteria culture parallel, this would be like if we added streptomycin to a live bacteria culture, saw them all die, and then concluded that, yeah, the cells don't have an antibiotic resistance gene that can protect them from the streptomycin. On the other hand, in the tube 4 of the lab group that did use kanamycin, we do see RFP. So this tells us that, yes, translation did work and protein synthesis did happen to make the RFP that we can see. That means the kinase DNA that we added, which again makes the kinase protein, was able to prevent the kanamycin from inhibiting the ribosome. So from this, we can conclude that the kinase from this antibiotic resistance gene added did protect our model from kanamycin specifically, and thus confers resistance specifically to kanamycin. Again, comparing it to a real cell culture, this would be like if we added kanamycin to live bacteria, saw them still live despite the presence of kanamycin, and then from that conclude that the cells must have some sort of antibiotic resistance gene that can protect them from the kanamycin. Again, while your students are only testing one antibiotic at a time, they will share the results with the rest of the class so everyone can see what happens with each antibiotic. As a reminder, in this lab, our first goal was to see if these antibiotics both inhibited protein synthesis. And the answer is yes, because when we added either antibiotic, no RFP was made, which tells us that protein synthesis was inhibited. Again, remember, although both antibiotics target protein synthesis, they each do so in their own specific way by binding to different parts of the ribosome. Our second goal was to figure out against which antibiotic the antibiotic resistance gene could act on and confer resistance to. And since we saw that when we added the antibiotic resistance gene, RFP was still made despite if kanamycin was added, and this did not happen with the streptomycin version. So we know that the antibiotic resistance gene confers resistance specifically to kanamycin. All right, I know I covered a lot. So here are the 3 takeaways again that I want you to make sure you at least remember. First is that each antibiotic has a different mechanism of action. The ones we looked at today specifically target bacteria ribosomes, but there are so many other antibiotics that target other processes. And even within a given target, such as ribosomes, there are specific ways they can be inhibited. Like again, the kanamycin blocks mRNAs versus the streptomycin blocking tRNAs from binding to the ribosomes. All right, that's takeaway number 1. Takeaway number 2 is that similarly, antibiotic resistance genes are specific in their mechanisms as well. For example, the kinase used in today's lab also had a very specific mechanism of conferring resistance. If you're curious, here's the nitty-gritty of it. The kinase actually alters the chemical structure of kanamycin, which is pictured here, and it alters it specifically at this red highlighted hydroxyl group. And when that happens, it renders the kanamycin unable to bind to the ribosomes to inhibit them. Now, on the other hand, if you compare that to the structure of streptomycin, you see that they have pretty similar chemical structures, but just there's a different group here. There's a different group here sticking out, kind of blocking that hydroxyl that the kinase would target. Because of that slight difference, the kinase can't alter it. And this is a great example of the specificity of the system. And lastly, the third takeaway is that our cell-free model with BioBits allows you to demonstrate this in a safe, easy way. Again, there are many different mechanisms that we could observe, but since BioBits contains bacterial ribosomes, it's the perfect model for us to specifically test the mechanisms of antibiotics that target protein synthesis, as well as antibiotic resistance mechanisms that show how you target those antibiotics. I know I talked a lot about live bacteria, but again, these BioBits cell-free models contain no bacteria, which makes it a very easy to use and safe system to look at antibiotics. So that is our antibiotics lab, our BioBits antibiotic resistance lab, how it's done, and the important takeaways that I'm showing here from the lab. Just to wrap up with some of our resources associated with this lab, if you're interested in the lab, all of our curriculum is actually free to download. Just go to the product page and click on the Downloads tab on our website. If you look up the BioBits Antibiotic Resistance Lab, it will contain the instructor guide with the full lab protocol. It will contain student guides with the background info, lab protocol, data tables, study questions, CER tables, and we'll also have classroom slides that are in PowerPoint format that allows you to edit them for your own use. And again, you can look at the link in the description of this YouTube video for the link to this lab to access this curriculum. And again, free to download. You don't actually have to buy the lab to access the curriculum. A couple of Q&A that were brought up when you guys signed up for the webinar. Some people ask how many students can use one kit. Almost all of our mini PCR bio learning labs, unless specified, have enough reagents for 8 lab groups. So if you put your students in groups of 2 to 4, which is what I recommend, one kit could cover anywhere between 16 to 32 students. Can students test both antibiotics? So I mentioned at the beginning that students will be split up, the lab groups will be split up, and half of them will test kanamycin and the other half will test streptomycin. Now, if you want your students to test both antibiotics, then each lab group will need twice as much reagents. So if you have 4 or less lab groups, like I said, all the kits have enough reagents for 8 lab groups. But if your classroom is only 4 lab groups or less, or you double up on the amount of kits, then yes, you could have each lab group test 2 sets of BioBits, 1 set for each of the antibiotics. That is possible, and that's outlined in the curriculum as well. Shelf life of the reagents. As long as the BioBits are stored properly in the freezer, then they will last at least 6 months. They usually last a little longer than that as well, but minimum of 6 months. Will there be more BioBits cell-free labs offered in the future? If you find that this cell-free model is really fun, we do have 2 other BioBits labs already on our website, and can't say anything too much, but yes, we are working on future BioBits labs, so sign up for our newsletter to stay tuned. And finally, someone asked, how does antibiotic resistance spread? So as I mentioned at the beginning of my talk, antibiotic resistance, the rampant spread of it has been largely due to misuse of antibiotics. And again, this lab does not cover the spread of antibiotic resistance. This lab covers the mechanisms or the specific ways of how it works. But if you are interested in covering this topic, we do have other labs that cover this idea. So showing you real quick, we have other related learning labs. If you want to do this lab, but you're worried about students not understanding protein synthesis or translation, you could scaffold this lab with our very first BioBits lab, which demonstrates the central dogma, shows how how DNA goes to RNA goes to protein. So if they don't really quite understand where the RFP would come from in this lab, this would be a nice lab to do first. And like I said before, if you want to cover the spread of antibiotic resistance, we actually have 2 other antibiotic resistance labs that actually end up pairing very nicely with the lab that I showed you today. Our first is our case study lab. This is an agricultural monitoring lab. This is a scenario case-based lab. We're using PCR and gel electrophoresis to determine if there's antibiotic resistance genes in soil samples in a fictional scenario. And then the non-fictional scenario is our eDNA project, where students actually go out, take samples of soils in the environment from farms, from their backyards, and come back and actually use, again, PCR and gel electrophoresis to determine if their soil samples contain bacteria with antibiotic resistance genes. So if you're looking to cover the spread of antibiotic resistance, or if you're looking for a next lab after you do today's lab, these 2 are great examples of that. And again, we will link these in the description down below as well, or in the chat. Please visit our resource page. We have tons of free educational resources. We have webinars like today's talk and lab demos on our YouTube channel. There's free worksheets and DNA Dots, which is our free readings. I mentioned Genes in Space up top. That is our free science competition where students can design experiments to send to space, and so much more. So please check that out. And finally, we have tons of other learning labs as well as resources at Mini PCR Bio. We have more BioBits labs. We have more labs that use the P51. We have labs that allow you to do PCR with our mini PCR, gel electrophoresis with our blue gel system. And if you are looking for an even cheaper way of running gel electrophoresis, you should check out our new Bandit system. It's gel electrophoresis for under $100. And then we also are coming out with more and more CRISPR labs, which are really, really fun as well. So thank you so much for listening to me. I really appreciated your attention. And if you have more questions, feel free to sign up for a demo here. This is a free one-on-one meeting with one of the team members, and we'll answer any questions you have about our labs, about ordering, whatever. So if you want to chat with us one-on-one, feel free to sign up there. Thank you so much for listening, and I look forward to hearing about how this lab goes for all of you. Thanks. Bye.